Hi, I'm David Potter with Fortis Life Sciences, and today I'm going to be talking about recombinant monoclonal antibodies and their use in research, diagnostics, and also disease therapy. So let's begin with an overview of the webinar. We're going to start by talking about antibodies in general and their suitability for use as tools. We will talk about monoclonal antibodies and how they compare with polyclonal antibodies. And then we'll move on to the various monoclonal antibody development technologies, dropping them really into two buckets, hybridoma and recombinant. And then we'll focus more on comparisons of recombinant monoclonal antibodies and hybridoma-derived antibodies in terms of kind of their intrinsic properties, how those properties translate into clinical suitability. And we'll also focus a bit on the ability to access diverse immune repertoires and what that enables. And then we'll end with a summary. So to begin with, antibodies they're really ideally suited for use as tools. When I see sort of a schematic of an antibody in my head, I see a wrench. You have the business end of the wrench, which fits whatever it's designed to fit, and a handle. And that's really what we have when we look at antibodies in terms of their variable domains, which are where the specificity of the antibody comes from. There's a very diverse range of specificities possible. We can have antibodies raised against pretty much any protein, post-translational modifications on proteins, which can include carbohydrates, also small molecules such as haptens. We can raise antibodies that can tell the difference between proteins that have mutations, even at a single residue. We can make antibodies against other antibodies. You're probably familiar with those in the context of secondary antibodies. Those will see antibodies of other species in general. And beyond that, we can even make antibodies that will see specific other antibodies. These are anti-idiotype antibodies that bind to the variable domains of other antibodies. And then you have the constant domain of the antibodies, which sort of constitute the handle, if you will. They provide a means for purification of many different antibodies using common methods, such as protein A or protein G affinity purification. They enable the detection of different antibodies, so it's possible to tell the difference between a rabbit antibody and a goat antibody, for example, based on those different constant domains between those species. There's also sites there for the addition of other molecules to the antibody. So you can conjugate on there, for example, HRP, or in the case of a therapeutic antibody, you can conjugate a toxin. Because those are going on to those constant domains, you're not affecting the binding specificity of the antibody. The FC defined by those constant domains also adds just bulk to the antibody, which is important for antibody pharmacokinetic properties, and also FcRn-mediated recycling in vivo. So if we take a look then at the different kinds of antibodies that are available, broadly speaking, we look at polyclonal antibodies and monoclonal antibodies. So I think this is the closest I come to an immunology slide in this entire presentation. And I think it's important to look at where antibodies are coming from so that we can understand the differences between polyclonal and monoclonal and the different ways in which we can generate monoclonal antibodies. So typically, the initial development of antibodies begins with an immunization, which elicits an antigen-specific immune response, and that immune response in the immunized subject is polyclonal. So it comprises many different monoclonal antibodies, which are themselves produced by monoclonal B cell populations. So if you're trying to make a polyclonal antibody, your material that you're harvesting is either serum or plasma, which contains the antibody itself. You can enrich for the specificity that you're looking for by affinity purifying on the protein or target of interest. You can even refine that specificity by absorbing out specificities that you don't want. One of the key things about polyclonal antibody production is that it always takes place in vivo. So anytime you want to make that polyclonal, you have to go back to animals, re-immunize and reharvest serum or plasma. Monoclonal antibodies, on the other hand, are they represent a single species subset from within that polyclonal population. And they're sort of defined by their source of origin, which is the B cell clone family that is producing that monoclonal antibody. And so in the case that you're trying to isolate and manufacture monoclonals, you're harvesting either the cells in the blood, or you're harvesting immune organs such as the spleen or lymph nodes. And then you isolate B cell genetic material from those B cells. And that genetic material is what's used to manufacture the monoclonal antibody, and that happens in vitro. Antibodies have many properties that lend themselves to use in research, diagnostics, and therapeutics. Ideally, these include the efficient and directed creation of the specificity that you're attempting to achieve, the ability to isolate that desired specificity away from other specificities that you do not want. Ideally, you would be able to produce the antibody in perpetuity. The behavior of the antibody that you're producing should be reproducible each time that you make it. You should also be able to make as much of the antibody at a time as you need. Ideally, the antibody will meet regulatory requirements, whatever those might be for the intended use. Preferably, you would be able to make changes to the antibody to suit the needs of the end use application. Ideally, you would also be working with a platform that can access the antibody repertoires of different species. So as we'll discuss later, different species have antibodies with different properties. And then ideally, you would also not have to use any more animals than are necessary, and preferably no animals at all. So if we look at those different ideal properties in the context of polyclonal and monoclonal antibodies, in general, you'll see that monoclonal antibodies are a better choice, with the exception of the cost to make them, where they cost more to make because the technologies involved are more complex. I won't go through this table in detail. We'll look at polyclonal antibodies briefly in the next few slides, and then we'll focus a lot more on hybridoma-derived and recombinant monoclonal antibodies. Some things I do want to point out are that high-quality polyclonal antibodies can have excellent specificity. So in some circles, polyclonal antibodies are a bit maligned. But as you can see here, we have an example of a polyclonal and a monoclonal against the same target where you really can't tell the difference in the Western blot application. The polyclonal has excellent specificity. It's also true that a poorly selected monoclonal can behave worse than a very good polyclonal. In addition to having the possibility of achieving excellent specificity with polyclonal antibodies, they're also known for having the ability to achieve broader specificities. So if we look at this Western blot on the bottom left, we're looking at an anti-human IgG heavy plus light antibody. We can see that it binds IgG1, 2, 3, and 4 heavy chains, and also kappa and lambda. If you were to try it to achieve a similar specificity with monoclonal antibodies, you would need at least 3 different clones to do that. The other thing with polyclonal antibodies is that because they recognize multiple epitopes on a single target, you have the ability to get increased signal potential. And that can be very important, again, especially in the case of secondary antibodies. A downside with polyclonal antibodies is that sometimes the nonspecific binding can be unresolvable. So here we have an example where we're attempting to make an antibody against a target protein using a peptide. There's some overlapping sequence with an off-target protein. And so when you affinity purify on that peptide from an animal immunized with that peptide, you'll get antibodies against a range of epitopes, which include antibodies that will bind epitopes that are recognized in the off-target protein. So this is an example of such a polyclonal antibody. If we look over on the right at the strip with a plus sign on it, we have a polyclonal antibody that sees the target at over 200 kilodaltons and has a couple of off-target bands. If we look at a panel of monoclonals raised against the exact same peptide, we can see specific monoclonal antibodies on strip 31 and 36 that see both the target band and the off-target band. And by not using those monoclonals, going with something, for example, like 21, strip 21, we have a monoclonal that doesn't have that off-target behavior. So that's a nice thing about monoclonal antibodies, is the ability to resolve some of the off-target binding that you see with polyclonals. So how are monoclonal antibodies made? Back in 1975, Kohler and Milstein published their seminal work on hybridoma technology. That took B cells, which are the antibody-producing cells. They are difficult to culture and short-lived in vitro. But if you fuse them with an immortal myeloma cell line, you end up with a cell called a hybridoma, which literally is the sort of the chromosomes of the B cell and the chromosomes of the myeloma cell jammed together into one cell. That cell is now immortal and also produces the antibody that the B cell used to produce. So that's traditional hybridoma technology. When we talk about recombinant antibody technologies, we're essentially talking about a family of technologies that result in the isolation of the DNA sequences responsible for encoding the heavy and light chains of the antibody. And then those are transfected into a heterologous host cell line, which then produces the antibody that the original B cell produced, but it's now a recombinant antibody. And so I've kind of grouped together three main technologies for doing this here. We have single B cell technologies, where the sequence encoding the VH and VL is directly cloned out of a single B cell. We also have phage display, which is generally performed on bulk B cell or peripheral blood mononuclear cell preps. And we can get the sequence of specific clones out through that process. And then there's a newer technology, which I like to think of as a reverse recombinant approach, which actually enables us to sequence the protein in an antibody preparation, either polyclonal or monoclonal, reverse translate from that protein sequence to a DNA sequence, and then transfect again into recombinant form. So if we look at the B cell approaches, there are many different ways to go about taking B cells and arriving at a recombinant antibody. This table from review by Pedrioli and Oxenius does a good job of comparing those technologies with hybridoma technology. They all have advantages and disadvantages. One of the disadvantages that we'll talk a little bit more about on a future slide with hybridoma is the amount of work involved on a per campaign basis. So you'll see that recognized there in terms of the red block for time and the red block for throughput. But if we look at the most commonly applied single B cell technology, that involves sorting antigen-specific B cells into 96 or 384 well plates, growing those, screening the output of those cells, the antibodies for suitability for an application, and then cloning and expressing the recombinant antibody. Another way that we can make recombinant antibodies is using the bulk B cell phage display approach. So this basically involves reconstructing essentially the whole B cell repertoire in vitro in a format that enables screening. That format is generally using a bacteriophage to display either a single chain FV or a Fab fragment on the surface of the phage. And then you have the sequence encoding that scFv or Fab linked to the scV or Fab through the phage surface display relationship, which enables panning for binders. So you pan the phage on antigen, you can adjust the stringency of the panning to get different levels of affinity or specificity. And then once you have a set of phage enriched through that process, then you would recover the full-length recombinant antibody. And then finally, as I mentioned, there's the de novo antibody sequencing, or the reverse recombinant approach. This doesn't require necessarily any cells. You're really starting with an antibody prep. This technology's come a long way now so that we can actually sequence the intact or the complete full-length heavy and light chain. There's some minor sequence ambiguity. It's still an expensive endeavor, and there aren't very many service providers that can perform this. And another disadvantage is that once you have that primary amino acid sequence, you can reverse translate that to more than one nucleotide sequence, and achieving good recombinant antibody expression can take some effort still. This can also be applied to polyclonal antibody as well as monoclonal antibody. In the case of polyclonal antibody, you're generally only able to recover the most abundant clones, and those aren't always the most desirable. In a polyclonal setting, there's also no information on correct heavy and light chain pairing, and so you have to mix and match heavy and light chains to try to identify the correct pairs. Okay, so what I'm going to do on the next few slides is talk more about the hybridoma-derived antibodies and recombinant antibodies, sort of in a head-to-head. The previous slides described the technologies, and it's important to remember when we talk about recombinant antibodies that they represent really the final product. The final product is a recombinant antibody, but they also reflect on the technology used to generate them. So hybridoma antibodies are all derived using hybridoma technology. There are nuances in terms of how that technology is applied, but recombinant antibodies can be derived from, as we just saw, several different technologies, and each of those can contribute in different ways to the properties of the sort of the end-to-end process of generating that recombinant antibody. So in terms of technology complexity, hybridoma is very well established. It's a fairly moderate complexity technology. Recombinant antibodies tend to be require higher technology complexity to achieve. Specificity isolation is very good for hybridomas, even better for recombinants. We'll see that on the next few slides. And as we go down the list again, we'll see unlimited reproduction is good for hybridomas, better for recombinants. Reproducible behavior, again, very good for hybridomas, better for recombinants. And then we start to really see these two diverge in terms of their suitability for scaling, regulatory acceptance, whether or not they can be engineered, their applicability across species, and the extent to which they use animals. So the whole point of having a monoclonal antibody is that it's monoclonal. Monoclonal antibodies should represent a single molecularly homogeneous prep of antibody. So it's important to realize when you think about hybridomas that B cell to myeloma cell fusion is a very low efficiency process. It's typically performed in the context of large numbers of polyclonal B cells that are fused in bulk. The expectation is that many of those B cells won't contribute to a successful fusion. And so after the fusion is performed, large numbers of cells are plated per well in 96 well plates. And then through a process of selection, unfused B cells and unfused myeloma cells are killed. And then you will end up with a well that contains supernatant, which if it's positive for binding to antigen in an ELISA screen, there can be three or more reasons why that might be the case. So 3 reasons I've listed here are that you could have an oligoclonal cell mixture, which includes a single antigen-specific clone. So in a perfect world, all you would have in there is a single species of hybridoma that is producing antibody against your antigen. In practice, what is very common in scenario number 1 is that you have a hybridoma in there that's producing antibody against your target, but you also have antibody in there, or a hybridoma in there that's producing antibody against a different target. And so your ELISA won't tell you that because it's only looking for the specific clone. It's also possible to have oligoclonal cell mixtures that include more than one hybridoma clone, both of which are secreting antibody against the same target antigen. From the ELISA data, all you'll know is that you have target-specific antibody in your well. You don't know that you have more than one clone in there. Also, again, because many B cells are fused with many myeloma cells in bulk, it is possible to achieve a 2 B cell fusion where more than one B cell is fused with a single myeloma cell. So typically with hybridoma technology, the way that monoclonality is achieved is through rounds of subcloning. So the cells are counted, plated out at less than 1 cell per well on average, and then screened again by ELISA. And so in the case of number 1, typically you would expect to see antigen-specific clones isolated from non-specific clones. So you would achieve monoclonality for the antigen-specific clone. In the case of mixed antigen-specific clones, you may very well achieve monoclonality of each hybridoma clone in different wells, but you won't necessarily be able to tell the difference. So you'll have individual subclones that are antigen specific, but it won't be obvious that it's actually 2 different clones. And then in the case of scenario number 3, where you have 2 different B cells fused with a single myeloma cell, that hybridoma is still a single cell. And so there's no way to resolve that by limiting dilution. You'll always have those 2 B cells contributing to the output of antibody into that condition medium. This was looked at in 2018 by Bradbury et al., where they enlisted the help of a few different labs working with hybridomas. They looked at 185 different hybridomas by next-gen sequencing. And what they found was that over 30% of those hybridomas, and these included hybridomas that are in common use for the production of antibodies against various targets across the industry. They showed that over 30% of these expressed at least one additional chain, either an additional heavy chain, an additional light chain, or both. It's a very interesting paper. I suggest that you go read it if you're interested. It does include rat hybridomas, which have a higher incidence of additional chains than mouse, so that skews the data a little. And it includes additional chains that came from the myeloma fusion partner. So some of the earlier myelomas that were used secrete or produce and secrete their own light chain. And so that can compromise things. Regardless of where the additional chains come from, it's important to realize that these additional chains can assort with the chains of the desired antibody. So if you have a monoclonal antibody that's being produced by a hybridoma, it has a heavy and light chain. If that hybridoma is also producing an additional light chain, the desired heavy chain will be able to pair often with both the correct light chain and the incorrect light chain. So you can get both a dilutive effect in terms of how much of your antibody is actually the correct antibody and is able to see the target. You can also see off-target behavior related to the additional chains. This really isn't a problem with recombinant antibodies. Recombinant antibodies start from isolated DNA for the heavy chain and the light chain, and typically those are maintained in bacteria where it's possible to pick a single colony for each chain and to confirm by sequence that what you're looking at is a single desired sequence. And then that's what's transfected into the heterologous host cell to produce the antibody. So there's really no issue with non-monoclonality when you're dealing with recombinant monoclonals. And the interesting thing in the Bradbury paper is that they actually made recombinant versions of the monoclonals from some of those flawed hybridomas. So they took just the correct chains and transfected those and compared those with the hybridoma antibody. And so what they saw was that the recombinant antibodies performed more cleanly and gave stronger, more specific signal than their hybridoma-derived counterparts, which is exactly what you'd expect from in a situation where you have additional chains being produced by those hybridomas. So another important aspect of using monoclonal antibodies as tools is that you should be able to go back and make them as often as you want. They should not be a limited resource. So this is true for both hybridomas and recombinant-derived antibodies. However, in the case of hybridomas, you're dealing with a physical material. So the hybridoma itself has to be stored. They're stored in liquid nitrogen or vapor phase nitrogen. In order to maintain those stocks, you often have to thaw them and expand them and then refreeze them. And so you have to deal with a physical inventory. Anyone who's done a lot of tissue culture or dealt with liquid nitrogen freezers may shudder a little. They've probably seen boxes with the edges missing and tubes floating in tanks. And so you certainly have to be very careful with hybridomas with maintaining those stocks and making sure that you don't lose that physical material. With recombinants, on the other hand, while there is a physical material, generally in the form of a glycerol stock of the plasmid expressing the heavy chain and the plasmid expressing the light chain, you also have the sequence as essentially data. And so that data can be archived in a secure fashion with backups. And so there's really no reason with offsite backups and with cloud availability that you would ever lose the ability to make that antibody. There is a great need for antibodies to be reproducible in the way that they behave. This can be more of an issue with polyclonals than with monoclonals, since polyclonals are at some level different every time you make them, since you're having to go back to a different set of animals and attempt to recreate the polyclonal that was made originally. However, even with monoclonals, I think there's a growing drive to make sure that these are recombinant so that they're all derived from a defined sequence. And having that sequence availability, as we just discussed, means that you can protect against loss of that antibody. You don't have to worry about your hybridoma becoming contaminated. You don't have to worry about genetic drift of the hybridoma cell line, so the accumulation of mutations through culture. These are all things that can affect reproducibility in research. And when you think about disease diagnosis and therapy, obviously the stakes are even higher than for basic research. And so the reproducibility bar is even higher. In order to take advantage of an antibody in some applications, it's important to be able to make a lot of that antibody. And so that means you really have to be able to robustly scale the antibody in order to meet need. One issue with hybridomas is that they are often finicky. So each hybridoma is really its own cell line. Even hybridomas from the same fusion will not behave in the same way. Often they will not expand in a predictable fashion. Sometimes you will expand the hybridoma and they will just decide to not grow past a certain point, or they may continue to grow so you can scale up the cell volume, but the level of antibody that they produce may change or decrease over time. The problem with hybridomas in that regard is that there's really nothing you can do about that. That's an intrinsic property of the hybridoma. Recombinant antibodies, on the other hand, can be produced and are produced at gram scales by transient transfection and at kilogram scales by amplified stable cell lines, which we'll talk about on the next slide. So again, the advantage of hybridomas is that they secrete the selected antibody. There's nothing else to make. You already have a way to produce the antibody. Recombinant antibodies, on the other hand, require a heterologous expression system. For small-scale transient expression, that involves preparing DNA in the amount needed for that scale of transfection. For larger scales, we would make a stable cell line. Those are expensive to make. It takes between 3 to 6 months to make them. And there's various technologies involved in making those cell lines, which may require outsourcing or even licensing. However, the advantage of stable cell lines over hybridomas is that you've separated out the selection of the antibody from the selection of the expression vehicle. So you're not stuck with a low-producing hybridoma. With a recombinant antibody, you can drive the expression in the way that you need to. And typically that's done using a host cell line such as CHO cells, for which there's an extensive track record in the production of antibody, and especially in the field of clinical materials. Of the top selling prescription antibodies in 2018, 7 out of the 10 were produced in CHO. There are many advantages of using cell lines such as CHOs, which includes their traceable provenance, so it's clear how that cell line came to produce the antibody that is going to end up in human patients. And there's a huge amount of knowledge around how to push good level expression from CHO cells. So that sort of segues us into the regulatory acceptance side of things. Hybridoma-derived antibodies are very well established in the diagnostic space. There are many go-to widely accepted hybridoma clones for common diagnostic targets. There's a level of trust of those hybridoma clones that is difficult to overcome because of the years for which they've been used. And there's generally not a problem producing the amounts of material that are required in diagnostic applications from hybridomas. So hybridomas do offer a good source of antibodies in the diagnostic realm. However, I think it is becoming the case that recombinant antibodies really are making inroads in the diagnostic sphere. Many of the newer targets don't have established hybridoma clones, so they're really in need of new antibody clones. There's also opportunities to improve upon existing hybridoma clones. Diagnostic manufacturers, as the number of those manufacturers increase and they're competing for access to clones, there's also a desire to secure their own supplies and to be able to own their own supplies. And so as the recombinant antibody technologies kind of are really driving primary antibody production in the industry, so we're starting to see recombinant antibodies contribute more in the diagnostic space. When it comes to therapeutics, I think there's some interesting numbers to contemplate here, where there's over 100 therapeutic antibody drugs so far that have received FDA approval since the first one in 1986. Upwards of 80% of those 100 plus drugs were developed using hybridoma technology, but only one of them is currently manufactured using hybridoma technology. All of the others are produced recombinantly. So there's a couple reasons for this. One is that hybridomas generated from wild-type mice yield mouse antibodies. Because they're mouse proteins, when they're injected into people as drugs, They have strong immunogenicity, which limits their effectiveness, their long-term use, and how safe they are. And so this is why only one currently of the 100 plus drugs are actually produced from a mouse hybridoma. There are now humanized mice, so these were developed in the 1990s. They produce fully human antibodies. Those fully human antibodies are minimally immunogenic, but you're still stuck with the manufacturing limitations of hybridomas in terms of their scalability and their long-term safety of those hybridomas as a renewable resource. So 60% currently of human therapeutic antibodies originated from wild-type mouse hybridomas. They're produced recombinantly, but their starting point was actually a mouse hybridoma. So that leads us to another major advantage of recombinant antibodies. Recombinant DNA technology enables antibody engineering. So if we look at a hybridoma, it secretes the antibody that was produced by the original B cell, and that's all it will ever secrete. However, if you clone the antibody from that hybridoma or from some other recombinant antibody input, you have a piece of DNA that encodes either your heavy chain or your light chain. And now you can apply recombinant DNA technology and modify that DNA in whatever way is needed in order to meet the end goal and arrive at a recombinant engineered antibody. And so the most common example of that when it comes to therapeutics is this process of humanization. So humanization enables the essentially the conversion of an originally mouse monoclonal antibody to a minimally immunogenic human monoclonal antibody. And that's done again using recombinant DNA technology, basically swapping out parts of that mouse antibody for human, starting with the constant regions, and then even within the variable regions, swapping out some of the residues from mouse to human through a process called CDR grafting to arrive at a humanized antibody, which is 90% human and far, far less immunogenic than a full-length mouse antibody. That's only possible through the use of recombinant monoclonal antibody technology, even with a mouse hybridoma input at the front end. So let's talk a little bit about some of the species that can be accessed through these different technologies. So really the founding species in monoclonal antibody technology was the mouse. That's where hybridoma technology was developed. They're a widely studied, widely used model of mammalian biology. There are lots of mouse models, lots of tools for working with mice. So they're a very popular starting point for antibody development. There's also a huge body of knowledge in terms of how to take a mouse antibody and humanize it, much more so than there are for other species of antibody. Some disadvantages, however, of mice are that it's difficult to break tolerance if you're trying to make an antibody against a mouse target. So as we said, there's lots of work done in mice, so there's a need for antibodies against mouse proteins to be able to facilitate that work, and it is difficult to make those antibodies in mice. From a manufacturing perspective, mice are small, and so they are not a good source for polyclonal antibodies. And so the polyclonal antibody industry really adopted different species, and we'll get to that in a second. Rat and hamster are also useful for producing hybridomas. There are rat myeloma cells that can be used to make rat hybridomas. And it's also possible to make heterohybridomas with using mouse myeloma cells, which I believe is how the first rat and hamster hybridomas were produced. They're not mice, so you have a bit of a better chance to make monoclonal antibodies against mouse targets in these species, although they are genetically quite similar to mice. rat and hamster really are much less used than mice for antibody production these days. Rabbits are also a commonly used laboratory animal. They're significantly larger than mice, so they have a larger blood volume, which makes them much more suitable for polyclonal antibody production. Originally, primary antibodies were predominantly produced in terms of large numbers of antibodies in rabbits as polyclonals. And so a lot of antibody manufacturers had large numbers of rabbits and a large body of knowledge in terms of the immunogens to use to produce those antibodies. And so those manufacturers looked for ways to be able to use rabbits to produce monoclonal antibodies. And so now you see in the industry a lot of monoclonal antibody producers using rabbits to produce these antibodies. And so that's really starting to drive diagnostic adoption as more primary antibodies are produced recombinantly in rabbits. Another interesting sort of sidebar with rabbits is that hybridoma technology was developed in rabbits, but it's essentially a proprietary technology which limited its adoption. And interestingly, even the holders of that IP now are switching to recombinant production over hybridoma. This table explains some of the differences between mouse and rabbit-derived monoclonal antibodies. Generally speaking, there are several advantages to using rabbits. The main advantage of mouse is, again, in terms of their track record of being able to progress a mouse monoclonal to a humanized therapeutic. But in general, rabbits, because they produce antibodies using a slightly different mechanism, actually have several advantages over mouse as an antibody generating species. And then finally, there are camelid antibodies. So it was discovered back in 1989 that camelid species produce a heavy chain only antibody. So these lack light chain. They contain a single variable domain, which recombinantly can be produced independently of the rest of the molecule as a very small 15 kilodalton single domain that's highly specific and can have high affinity for target and is also very stable. So these are really starting to grow in application. And one of the applications that really is quite powerful is the fact that it is much easier to engineer versions of antibodies that contain multiple VHH domains, greater than one VHH domain, than it is to try to make antibodies that contain multiple VH-VL pairs. And so this is enabling a new class of therapeutics called bispecifics. This sort of chart here from Brinkman and Konterman shows what's called the bispecific zoo. So this is really the next potential wave of therapeutics. Many of these formats are in the clinic now. This is really only achievable through recombinant antibody technology, being able to take different VH and VL pairs, or in the case of those camelid single domains, taking different VHHs and combining them into a single molecule to be able to bind more than one target at a time. It's going to be really interesting to see how these technologies play out in the clinic. And finally, animal usage considerations. Obviously, there's a big desire to use as few animals in research as can be reasonably used. Both hybridoma-derived and recombinant monoclonals are generally developed using animals. Their production usually does not use animals, although in the case of hybridomas, some of these are produced by ascites, which involves the intraperitoneal injection of the hybridoma back into mice. And so that can be a bigger usage of animals on the hybridoma side. Recombinant antibodies, on the other hand, can actually be derived using single B cell technologies from humans that have been exposed to pathogens. And in those cases, there's not any need to use animals at all. That's also the case in naive and synthetic phage libraries, where those can be reused over and over again for different targets. And again, that will reduce in the case of naive and eliminate completely in the case of synthetic phage libraries the need to use animals at all. So while animal usage can be eliminated from production with recombinant monoclonals, I would argue that we've yet to develop a technology that truly matches the antibody development power and efficiency of an intact immune system. So there is still a need to at least develop the antibodies in animals, but the production, the need for animals and the production of antibodies has really been eliminated at this point by recombinants. Okay, we covered a lot. So to summarize, polyclonal antibodies still meet important needs in the research and diagnostic marketplaces, especially in the context of secondaries. However, monoclonal antibodies provide for a limitless supply of a single defined molecule and all of the benefits that go along with that. The technologies associated with recombinant mAb discovery and development offer flexibility that you don't have with hybridoma. For example, you can't make hybridomas from people, where you can take blood from people that have been exposed, for example, to Ebola and find antibodies against that pathogen. So the antibodies themselves improve upon hybridoma-derived antibodies across many dimensions, as we saw in the comparison table. And they're really now starting to gain traction in the diagnostic market as more manufacturers are using recombinant antibody technologies to make their antibodies. And they have dominated and will continue to dominate in the therapeutic antibody market. I want to thank you for listening today. I hope you've found this helpful and interesting. Thank you very much.
Thank you very much, David, for a really interesting presentation there. It is now time for our question and answer session. So if you do have any questions for our speaker, there is still time to send those in. You can do that now. So David, the first question I have here for you today is, since you can clone antibody sequences from hybridomas, what are the main benefits of avoiding hybridomas to get all together?
Thank you for the question, Ellen. So from my perspective as someone who makes lots of antibodies, hybridoma is a good way to make a small number of antibodies against a small number of targets. But when you're really looking to industrialize the process of antibody discovery, the amount of work involved in performing a fusion and doing all of the tissue culture that follows from that, including multiple rounds of subcloning, it's really just much easier and simpler to start with a recombinant approach from the beginning. There are multiple pause points with recombinant antibody technology. It's just a much simpler process. Because of how inefficient hybridoma technology is, you also have to start with a lot of cells. And in a small animal such as a mouse, that generally means that you're going to have to take an immune organ such as the spleen or the lymph node, which means you have to sacrifice that animal. And if anything goes wrong with the fusion or subsequent steps, there's nothing to go back to. With the recombinant approaches, more often than not, and certainly what we do at Fortis, is we're starting with peripheral blood, mononuclear cells, and so in those cases where something goes wrong, or maybe you don't get enough hits from a particular animal, you can go back and you can get more start material because the animal isn't sacrificed during the process. So I would say from my perspective, those are the reasons to start with a recombinant process from the very beginning, whenever possible.
Fantastic, thank you. The next question here I have is, does it matter what cell type is used to express a recombinant monoclonal antibody?
So that's a good question too. Antibodies, full-length antibodies are complex. They involve fairly large proteins that fold into domains. They involve two different protein chains that have to come together. So the process of producing a full-length antibody is sort of complex at the cellular level. And so generally full-length antibodies have to be produced in mammalian cells. And they're the two sort of workhorses. We mentioned one in the talk, that was CHO cells. And then the other is HEC293s. So in the non-therapeutic realm, often folks will use HEC293 cells for expression. They're easy to work with. They grow really well. They produce good levels of antibody transiently. If you're looking at a therapeutic application, again, you're probably looking at CHO cells. Now for smaller fragments of antibodies, such as Fabs and scFvs, those don't necessarily need to be produced in mammalian cells. So there are Fabs that are produced in bacteria, such as E. coli, and the same for scFvs. Those sort of smaller, simpler molecules can be produced at scale in bacteria, and that can be cheaper.
Fantastic, thank you very much. I think we do have some questions coming in at the moment, so I'm just looking through those. The first one here I have is, what is genetic drift?
So we touched on that a little bit. It's the process of sort of mutations accumulating as you culture a cell. So it's really an issue for hybridoma. As we heard, you can have hybridomas that produce more than one chain. So sometimes you'll see the expression of a particular chain sort of go away over time with passaging those cells, or you can see mutations accumulate over passages of the cell, of the hybridoma cell culture. You kind of have to remember that B cells evolved to have a higher propensity for mutations within the loci that are responsible for encoding the antibody. And so sometimes that's a process that still kind of happens a little bit, essentially, in these hybridomas as you culture them. So it's the process of accumulating mutations which will change the nature of the antibody that's being produced over time.
Fantastic, thank you. And I've got another question here, which is, could you explain about cDNA insert into which cells, for example, E. coli or yeast or other?
Sure. So I think I talked about that a little bit in one of the earlier questions. So what cell line you're using, whether it's E. coli, yeast, or a mammalian cell, really depends. At the moment, it depends on the type of antibody material you're attempting to generate. The simpler, smaller fragments can be produced in simpler organisms, such as E. coli or yeast. It has to do with things like folding. It also has to do with things related to the glycosylation of the antibodies. So I believe in yeast, the yeast actually does glycosylate, and so you get you can get a molecule that's closer to a mammalian expressed antibody than you would, for example, in E. coli. Yeast, because they glycosylate, can also be engineered to glycosylate in particular ways. And so again, you can sort of tune how your antibody how your antibody will be produced.
Thank you very much. Just reading through the questions now. I've got another one here that asks, are you able to explain the affinity maturation procedure for antibody candidate sequence identified via phage library screening?
Sure, I'll do my best. It's not my area of expertise. So during the humanization process, as we mentioned, or as we discussed, one of the critical steps is the CDR grafting. So that basically involves taking these CDR loops, which are responsible for contact between the antibody and the antigen. They basically define the specificity. Taking those from the mouse antibody and grafting them into human frameworks. And so when you do that, very often things don't match up exactly right, and so maybe your loops look a little bit different in terms of how they're displayed, and so your affinity can be reduced during the humanization process. So you've made some changes, some pretty significant changes to the mouse antibody to make it look more human, and those changes often will have a negative effect on the affinity of the antibody. And so then affinity maturation is the process of trying to get your affinity back up to where the original mouse antibody was, or possibly even higher than the original mouse antibody. And that often is done by making back mutations, so changing some of the now human residues back to mouse. And you want to make the fewest changes back to mouse that are necessary to increase that affinity. And so that is usually done in the context of phage display, so that you can look at several different positions for back mutation, and then several different potential substitutions at each of those positions. So you would make a library of those various versions of the humanized antibody. And then through phage panning, again, you would use that stringency sort of adjustment to enrich for higher affinity versions of the antibody.
Thank you very much. That is all that we have time for today, but I really like to say thank you again to David Potter from Fortis Life Sciences for joining us and for today's really informative webinar. And thank you to everyone who's joining us online and for sending in your questions. I hope you've all found this to be a really worthwhile session. If you didn't have time to answer your question, or you have any more questions, we can follow up with these after today's event. Feel free to email me at editor@selectscience.net, and I will follow up with those questions for our speaker. You can also download a certificate of attendance for today's session, and you can find that in the related resources tab, which is to the left of your screen now. And if you'd like to listen again to today's webinar, or maybe invite a friend to listen, it will be available to watch on demand in just a few days' time. So for now, goodbye, and thank you once again for joining us.